Oh no. Is it recording now? Okay. Welcome to A&P. I am skipping to the slide where we needed to start. And so today is January 13th, Friday the 13th, and we are starting our lecture on slide 54. So sorry, it'll take me a second to get to that. One thing I've learned already, maybe I can get it to that slide before we start next time. Okay, did anybody get a chance to look at that handout talking about the gated channels, between the differences between sodium and potassium?
Yes.
Good. That's really important key is understanding the difference between the gates and the speed and how that affects generation of action potential and how the cell is able to come back to resting membrane potential. All right. So this is the slide we finished on, talking about depolarization, the whole phase, and we'll just start here real quick, but just as a reminder, in depolarization, our voltage-gated sodium channels, does it show up? Yes. Our voltage-gated sodium channels are, what would we say? They're fast. Why can't I write now? Forget it. Okay, so they're fast. I'm trying. No! Okay. And our potassium channels open slowly, and that is a really important thing to remember. So because potassium opens slowly, those sodium gates are open and sodium's rushing in, while the potassium gates are still taking their sweet, sweet, sweet time opening. In fact, they're still opening when the sodium gates finally slam shut. Okay? And because they're still opening and then you have those potassium channels open long after the sodium channels close, that allows potassium to basically move in and out of the cell to reach that equilibrium that helps bring that cell back to resting membrane potential. Okay, so repolarization. This is kind of what I was just mentioning, where those sodium channels begin to close. You can see that right here. But those potassium channels are still opening. Okay? And so, just as a reminder, if the channels are closed, there is no permeability to a particular ion. So when those sodium channels close, the permeability to sodium is almost zero, but potassium, the permeability is great because the channels are open. So it's the number of channels available that determine the permeability to any particular ion. And so because potassium is slow and it's still moving in and out, it helps, right here it says, notice, with repolarization, getting that cell back to resting membrane potential so that it actually can be re-stimulated again. Okay? Oh, and one quick thing, back to those gates. So, whoops, one too far. So, when potassium is moving in and out and causing the repolarization of the cell, the activation gates, remember we have two gates for sodium. We have our activation gates, and then we have our inactivation gate. Our activation gate for sodium, can I draw on the margins? No. So our why is this so hard? So our activation gates for sodium are to the outside of the cell. Okay. Oh, excuse me. So to the outside of the cell. And our inactivation gates are on the inside of the plasma membrane. Okay, I'm trying here. Activation. Oh, it won't let me write in the white. So activation gates on the outside. Inactivation gates are on the inside. And so during the repolarization phase, the activation gates close and our inactivation gates are open. So when our activation gates are closed and the inactivation gate is open, then you are back and ready to go again to re-stimulate the cell. So it has to be at this setting, basically, the gates open or close, activation close, inactivation open, in order for that cell to be re-stimulated. Okay. This is that handout I was talking to you about before. And as a reminder, I did upload a handout file with all those figures I mentioned that are not all in your textbook. This illustration is very important. If you're going to spend time studying any particular figure, this is one of the best because it talks you through each phase. So even when you're at rest, what are the gates looking like? So pink is your sodium, I'm trying, and the purple is our potassium, okay? So at each phase, resting, depolarization, repolarization, each phase, what's happening to those gates, okay? And that's something you will need to know. And so that's going to take you some time to go through and study, okay? So moving on to 57, we're going to talk about what after potential is. Okay, so many cells, after they've been stimulated, they're trying to reach resting membrane potential, but during that time, when you still are, after the cell has been stimulated and it's trying to reach resting membrane potential, there is a period where a lot of cells are hyperpolarized, okay? And so hyperpolarized, do you guys remember what that means? How would you describe it? So I see people going like this. So let's talk about the charge value of the resting membrane potential. Does it get closer to zero if it's no? Okay, so hyperpolarization means that resting membrane potential is even more negative, which makes it even more difficult or even impossible to generate an action potential during that after potential period. Okay? So it means that at that time, that cell cannot be stimulated because it is hyperpolarized. And the reason it's hyperpolarized is because those potassium channels are staying open and the potassium is moving in and out and it has not yet established the resting membrane potential. We know that potassium is very important in reestablishing resting membrane potential. We've talked about how you definitely need to know about the importance of potassium. And we have also talked about the sodium-potassium pumps. Sodium-potassium pump requires energy, uses ATP, pumps out how many sodium ions? 3. Pumps out 3 sodium ions for how many? Yes, so for 2 potassium that go in. So every 3 sodium that goes out, you have 2 potassium that goes in. So you have a net positive charge being pumped to the outside. And that also helps establish a nice steep concentration gradient for the sodium ions. And we've talked about why we need to have that nice concentration gradient. So for example, when those sodium gates open, you're going to get a flood of sodium in, and that leads to a graded potential. Remember that? And if the graded potential reaches our magical threshold, we have been able to generate our action potential. Okay? I know there's a lot of steps, but I think you guys can get this. Okay, then there is also what's called refractory period. Okay? And so if a cell is able to generate an action potential, which remember, action potentials have the ability to travel large distances across your body. Remember, they can go long distances. Graded potentials are localized. They cannot travel from one part of the body to another. Okay? Excuse me. Okay, so after an action potential is made, the cell, if you notice here, so the sensitivity of that area to be stimulated has decreased. So there's a period where it can't be stimulated. Okay? And so that we call that our refractory period. And there's 2 stages to your refractory period. Okay? So right here. So the first part, well, that's a 1. You have complete insensitivity, which means no matter what, you are not going to be able to stimulate that cell and generate an action potential. Nothing can make it do it. Okay? So complete insensitivity. Okay? And then you have, wait, hold on, let me double check something here. Boop. What's our next slide? Okay, so still talking about our first, the complete insensitivity. We call it absolute refractory. So that's the actual definition for the first period of this, okay, of your refractory period. You have absolute refractory, which means absolutely nothing can cause it to be stimulated. So let's see if Mike can draw my 1 nicer. I'm still working on using these styluses. Maybe I need a better one. Okay, so absolute refractory is the first phase. And so talking about gates, remember, so that we're talking about inactivation gates. So this is referring to our sodium ions, okay? So as long as your inactivation gates, what does this say, are closed, more depolarization can't occur. And of course I didn't bring my markers with me today. So remember, your inactivation gates for sodium are on the inside of the plasma membrane. Okay? So if your inactivation gate is closed, that means sodium can't move in or out. When the cell becomes basically reset, that inactivation gate will open and your activation gate is closed. So that is what it would be like at rest. When you're at resting membrane potential, the sodium voltage gated channels, the activation gate is closed and the inactivation gate is open. That is when a cell is ready to be stimulated again. But if that inactivation gate is closed, you're not going to be able to stimulate another action potential because no sodium can get through. Okay? So, let's see here. Then this next step right here, so when those inactivation gates do close and the activation gates are open, then it is possible to stimulate an action potential. But during the period here, when those activation gates are closed and they stay closed, not going to get an action potential generated no matter what. And so what's the point of this? I will just read this, so forgive me. It says, ensures that both depolarization and repolarization will be complete before you're able to generate another action potential. Okay? You have to reestablish your concentration gradients. If you don't, you're not going to be able to generate an action potential. And so these steps with the gates opening and closing and helping to reestablish that resting membrane potential is really important. And until it's reestablished, you can't do anything. Okay? So the second part of the refractory period, so what did we call the first part? What's number 1? Absolute refractory? Okay, so number 1 is oh my goodness. Wow. Okay, just forget that. Absolute refractory period. And then number 2, we call that a relative refractory period. And so absolute means nothing can happen at all. But the second phase, the last phase, that's our relative refractory period, which means that you could stimulate an action potential if you have a strong enough stimulus to reach threshold. okay? It has to be stronger than normal, okay? Stronger than the normal threshold. Because at this point, during this period of time, your cell is still hyperpolarized. So you need a stimulus that is stronger than normal to be able to trigger this hyperpolarized cell. Okay? And so if it is strong enough, you can generate an action potential. Okay? This stuff we've talked about before, so I won't just keep repeating ourselves. We know that there are channels that are open to potassium all the time. Okay, we've talked about that extensively. All right. Okay, so now we're going to talk about action potential frequency and different types of stimuli. And so how often can we stimulate an action potential? So the frequency, if we're talking about definitions, is just how many action potentials you can produce in a period of time. Okay? And so how many action potentials that can be stimulated or produced is, right here, notice, directly proportional to the strength of the stimulus and the size of the graded potential. Okay? So if you have a weak stimulus, you're probably not going to generate a lot of action potentials. But however, if you have a strong stimulus, which generates a strong graded potential, the likelihood of generating an action potential and more frequently is much higher. So a couple definitions for you that you're going to need to know: sub-threshold and threshold. So a sub-threshold, so first of all, what does sub mean? So I see people moving hands down. So when you hear that word sub, we're not talking about teachers, okay, not sub-teachers. Sub for me, what about the word subterranean? What does that mean? Below the ground, right? Yeah, so sub, you could think about sub means below, or a sub-basement is one that's partially below the ground. So this is what that prefix is talking about. So this is a stimulus that is below or not strong enough to make a graded potential. Okay? It's below the required strength. It's not good enough. Then the second one is threshold stimulus. This will actually produce a graded stimulus that is just strong enough to reach threshold. Okay? So this is like the minimum amount of strength or minimum amount of stimulus to generate threshold. If you hit threshold, you are able to have an action potential. Okay? So we want to remember that we can produce graded potentials. Graded potentials, if they're strong enough, reach that magical threshold level. Okay? If you reach threshold, what will happen? You'll generate what? Action potential, right. If you don't reach threshold, what happens? You just have a graded potential which stays localized, remember? So localized, it can't spread very far. Action potentials can. Okay. Okay, so some more terminologies. So, am I going too fast here? Do you guys have your notes online? Ooh, very nice. Okay, I always try to really emphasize the important parts, okay? So star the things that are really big deals. There are fill-in-the-blank notes online as well. And if you do have a question or you need to have me pause for a second, just let me know, okay? So a maximal stimulus. So these are all definitions right here. And you can find these in your textbook. So if you don't have time to write all the definitions down, just write the terms and you can look those up later. Okay? But a maximal stimulus, so this is because when we're talking about definitions, it's really hard to elaborate, so this is where I will read a little bit from the slide. I try not to read from the slides. So maximal stimulus is just strong enough to make, it says, maximum frequency of action potentials. And so a maximal stimulus is able to stimulate a lot of action potentials. So frequency is how many times an action potential can happen in a period of time. Okay, so frequency. So it's saying a maximal stimulus is able to produce the maximum amount of action potentials in a certain period of time. So you're generating a lot of action potentials. Okay, a sub-maximal stimulus, it's kind of silly. It says all stimulus or stimuli is probably a better word between threshold, which is our minimum that generates an action potential, and maximal stimulus. So sub-maximal stimulus is able to generate an action potential, but just not the maximum frequency of action potential. So it's everything in between. Okay? And then lastly, we have what's called supra-maximal stimulus. And so that's another prefix. Anybody taking medical terminology or have taken it? Yeah. If it's like a 1-credit class, I believe, and it's pretty helpful even after A&P. But it's a good class to take. Okay, so supra-maximal. And so this prefix supra is basically meaning, like, think about super or big. It's even more than a maximal stimulus. It's like, almost makes me think of Superman. But it's, so it's huge. It's a really strong stimulus. So notice it says any stimulus stronger than a maximal stimulus. So it's almost like supernatural stimuli, like just, whoa, super strong. Okay? And then this last little note down here. So it says the duration of absolute refractory period. So as a reminder, absolute refractory period is the first phase of the refractory step. Remember, we have 2 phases. And so in absolute refractory, are we able to generate an action potential? No. Very good. Okay, so during this period of time, it's going to determine the maximum frequency that a cell can generate when it's excited. Okay. Oh, I haven't been watching the time. Oh, we're doing good. I know. It's going to be okay. It's going to be okay. Stretch the fingers. Move the wrists. Okay? All right. Propagating action potential. So we have spent a day and a half of lecture talking about how we actually are able to generate an action potential. Now, we're going to move to talking about, okay, we've made an action potential. How do we get that action potential to move from one place to another? How do we get it to move from one part of the body to another part? How do you get that signal to move? So that's what we're going to talk about. That's what it means when it says propagation of action potentials. It is getting it to move and continue to send that signal. Okay. So action potentials, as it says, do spread across the plasma membrane. It's different than the idea of a graded potential. Remember, we kind of described a graded potential as like a drop in the water and you have that ripple spreading out. Very nice. But with action potentials, which are occurring along the axon, we have a little bit of a different arrangement, and that's what we're going to be talking about. And so action potentials, they are produced on a location on the plasma membrane, and it will stimulate the spot right next to it. So it is similar in how graded potentials, so let's talk about, remember we have, okay, let's say we have one channel, this is a little bit of review, one sodium channel, and it opens up. Because it opens up, it stimulates the channels right next to it to open, and stimulates the channels right next to that. So you get this positive feedback cycle, which causes all those channels to open, and sodium rushes in. Okay? So remember that idea. The same kind of thing happens with action potentials and how it's going to be able to send the signal. Action potentials are kind of neat because they only travel in one direction. Okay? Gradient potentials spread out. Action potentials only go in one direction. Right? But it's still the same idea. If we have a channel open that's been stimulated, it will stimulate the one right next to it to open and cause an action potential. And so it will do that same kind of wave thing, but in just one direction. Okay? And how that action potential spreads down an axon, notice here, depends on whether it is myelinated or unmyelinated. And we'll talk about both of those and how that signal is able to pass. Do you remember what myelination is? Or it's wrapped in what? Yes, okay, so what cells, okay, there's two cells that are able to myelinate an axon. Oligodendrocyte, I heard that, and Schwann, okay, both of those cell types, they wrap their plasma membrane around and around and around and around that axon, and so it insulates it. Now, where do we find oligodendrocytes? Central nervous system, and then where do we find our Schwann cells? Peripheral. You guys are awesome. Okay? Excellent. You guys are remembering so many things. So first, we're going to talk about how an action potential spreads on an unmyelinated axon, because it is different than how it spreads on a myelinated axon. Okay? And by the way, if this video thingy works, I will upload it to YouTube and create a link for you right away so you can even look at it later today. So that's the goal, is that you guys will be able to rewatch and listen to lectures and give me some feedback on how that works for you with your studying. Okay. All right, back to the unmyelinated axon. So it's just an axon. There's no insulation. Because remember, myelin insulates that axon. And insulation is really helpful if you're trying to contain an electrical signal and keep it from, it helps it speed up. Okay, so with an unmyelinated axon, notice it says it basically stimulates the area right next to where that action potential first was stimulated. These only go in one direction, okay? So if this is the plasma membrane, okay, first of all, what is the charge right on the inside? What should it be? Negative, right? Ooh, I can finally draw a little bit. And then right outside is positive. And so what happens with an unmyelinated axon, if we get a stimulus right here and we have depolarization, ooh, that's so cool. The charge switches, right? And because it switches, it then stimulates the spot right next to it, and then causes the same thing to occur. It causes, it causes the charge to switch there as well. And then it goes to the next spot, next thing, next thing. So it's kind of like the wave. And as you stimulate the next area, the one behind it is going through repolarization, and it has that refractory period, the absolute refractory period, where it cannot be stimulated anymore. And because of that, that is why the signal only can go in one direction. Okay? That's the key. That's why it goes in one direction. And so you have a depolarization. It stimulates the next area while that one here undergoes repolarization. and so on. And so as soon as the charges switch, it triggers the next spot, and then the previous ones are repolarizing. Okay? And so we call this, because it's traveling along the entire length of the axon, we call it an ionic current. Okay? So notice says if an action potential is initiated at one end of an axon, I accidentally erased my little line here, at one end of an axon, it is only going in one direction. And so remember, it can only travel in one direction because when the area previously is trying to reestablish its resting membrane potential, you have a period of time where it cannot be stimulated no matter what. And so it can't be stimulated, and you have the next area that has depolarized. And then it moves on over here, and so this next spot is undergoing repolarization and cannot go back and trigger the previous area because it's an absolute refractory. And that's the key to going in one direction, which is the name of a band. Sorry. Okay, so as I have already said, notice right here, it says absolute refractory period ensures one-way propagation of action potential, which is what you want. What if you generate an action potential and it starts traveling one way, but then you're able to re-stimulate the ones behind it? What happens to your signal? Where does it go? Your neuron's going to be super confused. The signals are not going to get to the places they need to be. You're basically going to have a lot of confusion back and forth. So it's really nice, this absolute refractory period, and it makes it only travel in one direction. Okay, here is a lovely illustration. This is in the handout that's provided on Moodle. I'm not sure if this exact picture is in your book, but one very similar to it is. And this is illustrating the steps in the process that occurs in this, the propagation of action potential on an unmyelinated axon. Okay. And so this actually, we have 4 steps. It's a good picture. This represents right here a section of, oh, stop it. How do I make that go away? Okay. This represents a section of the axon, not myelinated. It shows you the charges, so negative on the inside, positive outside. This is resting membrane potential. Okay, there it is unstimulated. But if you get a stimulus in a certain area, okay, you cause depolarization, and I love it. They show it right here, depolarization where the charges flip, because that is what happens, okay? And with this, because we have that absolute refractory period, I know I've said it a bunch, that only allows it to propagate in one direction. So as it stimulates the spot right next to it, right here, the area behind it is trying to reestablish resting membrane potential, which then causes it to slowly move down one direction, down that axon. Now, because it's not myelinated, how will it affect the speed of the propagation of the action potential? Yeah, it's going to be slower because it has to go through every little spot here along the way. So there's nothing it can skip. It has to go the entire length. Okay, and this is really like someone doing, or like a group of people doing a wave in a stadium. Okay, because you have to have the person before you do the wave, and then because they did it, and then you do the wave, and the person next to you does the wave. Ooh, the whole line of people have to do it. We don't get to skip people. Oh, I don't know if they do the wave very often anymore in coliseums, but they're so fun when everyone does it. Have you guys ever been to like a game, like a baseball game?
We did it like 2 seasons ago. We had the season record for one.
For doing the wave?
Yeah.
Really?
Yeah.
Which game were you at?
It was always one by the Wisconsin one. It was the Wisconsin game.
Wisconsin? You were is there a team in Wisconsin? There's a baseball team?
Oh, gosh. No. Huskers football.
Oh, for football. Okay, cool. Sorry, I'm more of a baseball person. But football is cool too. Okay, now we're going to talk about how an action potential can travel down a myelinated axon. Woohoo! Right? Okay, but we already know that because it's myelinated, how does it affect the speed of how that signal travels? Faster! And you know how it's faster? Or why it's faster? It's because of these nodes of Ranvier. You have to say it with a French accent. Okay? Nodes of Ranvier. I used to say Ranvier. No, it's actually Ranvier. So these nodes of Ranvier are those little gaps in between the myelin. So here I'm going to draw this little line. That's our, what is that? Oh, just blanked on the name. Axon. Okay, that's our axon. And then we have, so we'll pretend that we're in the peripheral nervous system. So what cell would this be that's myelinating in the peripheral nervous system? Schwann. Oh, come on, I'm trying to spell Schwann. Okay, good. And then we have another one. And another one. And we have these little gaps in between. Well, those little gaps in between, those are our nodes of Ranvier. Okay? And so what's really neat, you have those gated channels, those sodium gated channels that we've talked about, and they're highly concentrated in these areas of nodes of Ranvier. And so because of that, it allows the signal or the action potential that's generated to jump from one node to the next, instead of having to travel the entire distance that you have to do on an unmyelinated axon. Okay, so because it can jump from one to the next, that makes it faster. And so that type of propagation where it jumps between these nodes of Ranvier, we call that saltatory conduction. And so what did we call it when it had to travel along the entire length of an unmyelinated axon? Do you remember? It's the ionic current. Okay, so ionic current for an unmyelinated axon has to travel the entire length. When it's myelinated, jumps from node to node to node, we call that saltatory conduction. So different ways the signal is sent, and with saltatory conduction, it's faster. Okay? And so let me see, make sure I didn't miss anything. Okay, so we already know that a myelinated axon is going to be able to propagate a signal faster than one that's not, but there are some other things that can influence the speed of the action potential. So right here, down here at the bottom, the speed of the transmission is also affected by the thickness of the myelin. And so, it's right there. So the thicker the myelin sheath, the faster the signal is sent. Okay, so if we're comparing two myelinated axons, one with a thinner myelin sheath and one with a thicker myelin sheath, the thicker one's going to send it faster. Okay, so thicker or larger in diameter, so we use a diameter right here. So that is another factor that influences the speed. Not just whether it's myelinated or not, but also the thickness of the myelin sheath. Okay. So here's a nice figure showing saltatory conduction. This is where we have our nodes of Ranvier. And as a reminder, in these little gaps, these nodes of Ranvier, we have a concentrated amount of what? Do you remember? A bunch of a certain thing here. Our voltage, oh, come on, voltage gated sodium channels, okay? So way more concentrated than you would find on an unmyelinated axon. And so it's highly concentrated, so that helps you to be able to have a really big depolarization event So you have generated a strong action potential that can then jump to the next place. And as this place is getting stimulated, the previous one is going through repolarization and refractory period. Okay? So again, the signal is only able to travel in one direction. So no matter what, if it's myelinated or unmyelinated, the signal can only go in one direction. And I really just can't stop thinking about the band every time I say that. Okay? All right. So, we've moved past action potentials. Yay! We've talked about ionic current, how it's propagated, how it's spread. So that's ionic current on an unmyelinated axon. We've talked about how it's propagated on a myelinated axon. So once the signal that is propagated down an axon reaches the end of that axon to the synaptic terminal, those little buttons at the end, then we get to talk about what happens there at the synapse. And we've talked about the synapse with the muscular system, and so some of this will be review for you, which is nice. A little bit of a refresher. Okay, so at the synapse, this is basically, as it says, this says junction, or the place where you have two cells close together that are going to be able to interact, okay? They're going to be able to communicate with each other. And when we're talking about nerves, the communication method is going to be with neurotransmitters. We've talked about a neurotransmitter before with muscles. Do you remember what that one was called? Starts with an A. Oh, I heard someone whisper it. Acetylcholine. That's a neurotransmitter. That's how the neuron communicates with the muscle cell. Okay? So we're going to talk more about this fun stuff. So when we're talking about synapses, we have a presynaptic cell and a postsynaptic cell. If we use the muscular system example with acetylcholine, let's see, the neuron there's our little synaptic node right there, and then here's the postsynaptic membrane. Okay, so this is the presynaptic cell, and this is our postsynaptic cell. Okay, and so the presynaptic cell is the one that's going to release neurotransmitters into the synapse. The synapse is this little gap here. Okay. Right. Do we have time? It's 48, 49. I'm trying to just... Oh. All right, guys. We'll stop here and start on Monday on slide 68. Please spend some time studying this weekend and enjoy your long weekend as well. Thank you very much. Now, how do I stop recording?